Hello, everyone. My name is Harrish Ganesh, and I'm a rising senior at Thomas Jefferson High School for Science and Technology. And today, I want to discuss the question with you: will molecular docking experiments lead to a potential HIV drug discovery and for other diseases as well? So, just a little bit about myself. I'm deeply devoted in virological and neuroscience research, and I love to spread my ideas and knowledge. For example, through the TED Ed Club I started at my school. And I love to play a variety of sports, such as basketball and football. And I'd like to thank the Johns Hopkins Global Health Team for giving me this opportunity to present. And I'd also like to thank my school principal, Dr. Bonatadis, and my teacher, Dr. Mora, who are always there to help and support me. So let's begin with an overview. First, we're going to talk about some little bit about background of HIV, and then talk about the drug synthesis process. Then we're going to transition to molecular docking and its applications, and how I used it in my research, and maybe how you can as well. So you may be wondering, what is HIV? And the human immunodeficiency virus is HIV, and it's infected through many ways, such as unprotected sexual activity, sharing injection equipment such as needles, mother to baby during pregnancy, and even after pregnancy during birth and breastfeeding. And also, you can get it through contaminated blood transfusions. And there's a little fun fact for you on the right. So, what's the issue with HIV? Is that HIV affects millions worldwide. Nearly 37.6 million people globally were living with HIV, and 1.5 million people became newly affected with HIV in 2020. This led to 690,000 deaths from AIDS related illnesses that year. And millions of people need adequate medication to combat this deadly virus, as 27.4 million people were accessing antiretroviral therapy in 2020. But what makes HIV so difficult to treat, you may be wondering? And it's because HIV is a retrovirus. And in simple terms, this is just any sort of virus that translates and converts its RNA genome into a DNA copy. And why? Because the host cell that it's infecting has DNA. So it translates its RNA genome to DNA to insert it into the host cell's genome. And it can't be removed from the cell unless you kill the cell. But if you kill the cell, you're going to be releasing other viral content, and that viral content is going to infect more cells, making matters worse for the patient. But if there was a drug available to eradicate this disease, how long would it take, you may be wondering? Well, there's a long road to a new drug, right? A drug that enters a market takes decades and decades to arrive. From start to end, the process can take up to 15 years to complete, as the idea, synthesis, and testing and data analysis is one that requires great scrutiny before it's released to the market. But unfortunately, creating a drug to become available to the public is not a simple task. It requires millions and millions of dollars. The testing of a variety of drugs with a large amount of money due to that can lead to very few amount of drugs becoming released on time to help the millions of struggling of patients. So let's go ahead and talk about the different drug approaches and how they discover them. So the first one is called serendipity. What does this mean? It just means luck. So oftentimes drugs are not meant to be found, they show up themselves. So for example, in 1928, Sir Alexander Fleming studied Staphylococcus. And during the studies, there was a contamination of plates with airborne mold. And he noticed that this mold were actually killing the bacteria. So he used this mold to lead to the antibiotic penicillin. Pretty interesting. Now, there's also screening. So, screening is when you have an enormous amount of chemical compounds that are tested to see its resulting biological effects. And basically, what happens is that while this has been previously successful, it's a trial and error process. So, because it's really trial and error, it can be very costly and time consuming. And thus, it doesn't have a clear structure. A more traditional method is chemical modification. And chemical modification is when you slightly change a compound to improve its efficiency. So you often leave with a molecule that is very similar to what you started with, but it has shown to work in the past. But now, the one that I want you to focus on the most is mechanism-based docking approaches. And this is when the interactions between various proteins are analyzed to see its interactions and how they can disrupt the disease process. And this is an extremely yet somewhat overlooked portion of molecular docking, as molecular docking is a huge advantage to this type of research. But what is molecular docking? So molecular docking, in simple terms, is just a computational process in which two proteins are analyzed to see its most likely binding conformation in the natural process. So it's a computer simulation in a way where you can see how two proteins may interact inside a body. And this orientation can be used to improve drug binding testing and its efficiency. So this, you may be wondering where this is heading. So if you're still having a little trouble visualizing this, think of it as a lock and key, where one wants to find the correct relative orientation to the key, which will open up the lock. So during the docking process, the proteins being studied must adjust their conformation to achieve an overall best fit. And you may be wondering, why would we want to use a molecular docking experiment if it's just a computational procedure? Well, the biggest advantage is that they're extremely fast and affordable for researchers, enabling them to test proposed compounds, drugs, and proteins for its efficiency, improving and getting rid of a disease mechanism. And this is extremely important because you can get drugs faster in the market. So molecular docking experiments are then connected to a wet lab research. And a wet lab research is just in person in the lab, in the laboratory research and testing actual compounds. So by analyzing a computational interaction between two proteins and comparing that to the lab, you can save so much time by simulating these proteins. And by simulating the proteins, you can see the interactions faster and be able to do more work faster, saving time and therefore money, and therefore being able to get that drug out in the market faster and more than ever than before. So what does this mean we can do with molecular docking? Well, did you know that that COVID vaccine that you probably already have right now had actually been used by molecular docking experiments and researchers to see how it affects the human body and how it interacts with proteins? And that vaccine itself was able to get out to the market faster during such an important time such as this pandemic in order to help the millions of patients around the world with COVID. And also the HIV vaccine. While there isn't a vaccine yet, there's a lots of antiretroviral therapy and drugs trying to be produced to be able to create a vaccine. And molecular docking is a great way for researchers that were able to use it to be able to create compounds to reduce HIV side effects. And those drugs that you see in your local pharmaceutical store, those drugs are also been used by molecular docking researchers that use these drugs and proteins and analyze them to proteins in our body to see how they may interact and affect a sort of mechanism in your body that's creating a sort of pain, let's say. So what did I use HIV in molecular docking with? So I combined the two. So molecular docking is extremely accessible. Even I can go ahead and use it. And basically what I did was I analyzed a human receptor cell and the HIV virus glycoprotein. And I want to see which part of the glycoprotein of the virus most likely binds with the receptor, which loop, which amino acid loop. And I found that the V1 loop through these simulations over 2 years of research, that the V1 loop of this glycoprotein, it's like a strand of the protein, actually interacts with the with the alpha 4 beta 7 lymphocyte receptor, as you're seeing here on the right. Pretty interesting. So what should you take away from this? So understanding the interactions between proteins is key for drug discovery effects, and it's extremely successful in discovering drugs that were to treat cancer and other diseases such as malaria. So this is a relatively new technology in which silico computational techniques can be used to analyze and predict molecular chemistry in an inexpensive manner. And even you can take a role in adapting to molecular docking research in your work. And that's the great thing about this. It's accessible to everyone and you can make an impact on society. So once again, I would like to thank the following people: Dr. Ann Bonatadibus, my principal, and Dr. Kathleen Morrow, my biology teacher. And here are my citations. And thank you so much for listening, everyone. I hope you had a great time and feel free to email me with any questions. Thank you so much.